In this video, guidance will be provided to perform a batch activity test to assess the biological sulphate reduction process. Microbes play a major role in the biogeochemical cycle of sulphur. The oxidation states for sulphur are -2 for sulphide, 0 for elemental sulphur, and +6 for sulphate. Wastewaters containing high sulphate concentrations can cause an imbalance to the natural biological sulphur cycle by altering the biodegradation pathways and the kinetic rates. The sulphate oxidized from sulphur in the atmosphere can be deposited in the wet or dry form. Depending on the redox conditions, it can be reduced to sulphide, which in turn can be oxidized to sulphur or sulphate by microorganisms. However, during anaerobic wastewater treatment, sulphate can be reduced back to sulphide by sulphate-reducing bacteria, also known as Srb. Assimilatory or dissimilatory sulphate reduction are the most important anaerobic processes that affect the conversions of sulphate. The initial step of biological sulphate reduction involves the transfer of exogenous sulphate through the bacterial cell membrane into the cell. From both processes, the most important is the dissimilatory pathway. The dissimilation process proceeds via the adenosine triphosphate ATP sulfurylase after reaching the cell membrane. ATP produces the highly activated molecule adenosine phosphosulfate APS and pyrophosphate PPI. The APS is rapidly converted to bisulfite HSO3 by the cytoplasmic enzyme APS reductase. Pyrophosphate is hydrolyzed, and the sulfate of APS is reduced to bisulfite, which in turn is reduced via a number of intermediates to form the sulfide ion. The sulfate-reducing bacteria, Srb, are facultative anaerobes that live in oxygen-free or oxygen-depleted environments. They utilize sulfate as a terminal electron acceptor to produce hydrogen sulfide, H2S, as one of its metabolic end products. Srb are frequently present in sewerage systems and wastewater treatment plants. They are also present in freshwater, marine, hypersaline, and oil or hydrocarbon polluted sites. The microorganisms responsible for the reduction of sulphur compounds belong to both bacteria and prokaryotes. In general, the bacterial sulphate reducers are characterized into different branches. The delta proteobacteria with more than 25 genera and different species of gram-positive and gram-negative bacteria. Sulphate present in wastewater is the electron acceptor, while Srb can oxidize a wide range of substrates as the electron donors. These include, among others, hydrogen, alcohols, fatty acids, aromatic and aliphatic compounds. In the present video, the Srb activity, Srb ANA2 test, will be assessed through the execution of a batch activity test. The experimental data will be used to estimate the volumetric rates of sulphate reduction and sulphide production. Different kinetic parameters of interest will also be determined, namely the specific volatile fatty acid consumption rate and the specific sulphate uptake rate. Specific precautions need to be taken during the execution of this batch activity test. In general, strict anaerobic conditions should be maintained by avoiding the intrusion of oxygen. Adequate mixing should be provided to keep homogeneous and well-mixed conditions and also the biomass in suspension. Because Srb are highly sensitive to pH and temperature, they should be maintained at the desired set points and also be monitored as often as possible to avoid potential deviations from the desired ranges. To determine the stoichiometric and kinetic rates of interest, samples should be collected periodically at the corresponding sampling times from both the gas and liquid phases. In this video, the batch activity tests will be performed in serum vials, fitted with a septor, and sealed airtight with aluminium crimps. Both gas and liquid phase samples will be collected and analysed for different sulphur species. The batch activity tests will be performed following a sacrificial mode, and therefore several serum bottles are required to perform one test under specific conditions. The detailed demonstration of this activity test will be shown in this video. In general, samples should be collected once every 5 minutes, during the first 30 to 40 minutes of duration of the batch activity test. This is similar to certain methods described previously, like for the EBPR activity tests. Liquid samples should be collected by inverting the vial, and sufficient care should be taken to avoid the intrusion of bubbles in the samples. In the sacrificial method to perform the activity tests, the samples should be collected in duplicate. A defined volume of gas should be collected from the headspace of the vials, using airtight syringes for hydrogen sulfide analysis. The samples should be collected in duplicate. The analyses of the gas samples should be performed immediately after the collection of the samples, to avoid volatile losses. This working sheet can be used as a guide and example to prepare and execute the SRB activity test. In the following video, the main steps to carry out the SRB activity test will be demonstrated. It includes the description of the apparatus, sludge preparation, preliminary activities, as well as the method for the collection of samples. It also shows different activities that need to be performed before, during, and after the completion of the test. Collect all the materials and equipment required for this test. Prepare the correct volume and strength of the phosphate buffer and sodium hydroxide solution. The DO, pH and redox sensors should be pre-calibrated. Also confirm that your working sheet is ready. Syringes, stopwatch, shaker and all materials should be placed next to your setup. To avoid cross-contamination, use new disposable syringes, filters and disposable two-way plastic valves. This test will be performed in a sacrificial mode. Hence, sufficient serum bottles and sampling cups will be required. Transfer 2 litres of activated sludge in a beaker and homogenate the sludge by mixing. In this case, the sludge was mixed for approximately 5 minutes. Select an appropriate mixing speed to avoid the accumulation of biomass at the bottom. Once proper mixing is assured, the initial parameters such as pH, redox, dissolved oxygen and temperature should be recorded. Rinse the pH electrode with water and wipe it with a soft tissue paper. Place the electrode firmly inside the activated sludge and wait until a constant reading value is reached. Rinse the electrode properly with water and switch off the sensor. Thereafter, measure the temperature and the DO. Rinse the electrode carefully with water and wipe it with a soft tissue paper. Immerse the sensor in the activated sludge and wait until the reading values are stabilized. Record the temperature and DO values. Clean the electrode properly for further use. Check the concentration of nitrate in the activated sludge using nitrate strips. Take a clean strip from the container and dip it in the mixed sludge for 2 to 5 seconds. Remove the strip and shake off the excess liquid. After 1 minute, compare the colour of the strip with the colour chart provided. Finally, measure the redox potential of the sludge sample. Rinse the electrode properly before use. Immerse the electrode properly in the mixing sludge. Hold the electrode for 15 to 20 seconds until the ORP readings reach a constant value. Rinse the electrode properly with water and keep it for further use. Read the procedure carefully and make sure that the materials required for the collection of samples, such as sampling cups and syringes, are available. Immerse the plastic syringe in the mixing sludge and move the plunger up and down at least 5 times in order to collect 5 millilitres of a homogeneous sample. Transfer the sample into a labelled sampling cup. Collect the samples in triplicate by following the same procedure for sample collection. Close the lid of the sampling cups as firmly as possible. Store the samples for mixed liquor suspended solid and mixed liquor volatile suspended solid analysis. First, prepare the materials required. Take a dry 110 milliliter serum bottle and place it on the weighing balance. Zero its weight. Fill the bottle with demi-water. Fill the bottle up to the top. Write down the final weight of the filled bottle. This volume corresponds to the actual volume of the serum bottle. Take a dry 110 milliliter serum bottle on a weighing balance and zero its weight. Using a clean plastic syringe, collect the sludge from the beaker. Slowly add 75 grams of sludge into the serum bottle. Avoid any air bubbles during the transfer of the sludge. The weight of the mixed liquor from a conventional activated sludge system is comparable with water and thus can be considered to have a density of 1 gram per milliliter. Write down the weight of the bottle on the working sheet provided. Take the already prepared phosphate buffer solution. Transfer 3 milliliters of the phosphate buffer solution to each serum bottle. Write down the final volume after the addition of the phosphate buffer solution. Close the bottles with the rubber stopper. Use aluminium crimps and a vial crimper to seal the serum bottles airtight. SRB activity tests should be performed under anaerobic conditions. Take the serum bottle containing the activated sludge and the phosphate buffer solution. Connect the water lock to the serum bottle using a needle arrangement. Insert the needle that connects the nitrogen gas tubing into the serum bottle. Open the nitrogen gas supply and purge the bottle for 1 minute. Thereafter, stop the nitrogen gas supply and wait until the bubbles in the water lock cease. Disconnect the tubes from the bottle. Place the bottles on the orbital shaker. In this example, a speed of 200 revs per minute was used in order to avoid the sludge settling. The SRB activity tests are carried out for 360 minutes. They are conducted following a sacrificial mode approach using duplicate bottles. Place all the bottles on the shaker for 20 minutes. After 20 minutes, stop the shaker. Remove the bottles from the shaker to collect gas samples. Use a 5 ml plastic syringe fitted with a two-way valve. Insert the needle into the headspace and push and pull the plunger back and forth to flush the syringe and collect 3 ml of gas sample from each bottle. Close the valve to prevent the loss of the gas sample and analyse the gas samples as soon as possible. Samples collected for the determination of sulphide should be preserved in sodium hydroxide solution, as described in the textbook. In addition to its composition, it is also recommended to determine the gas volume and to measure the pressure of the bottles, as explained in section 4.7 of the textbook. Repeat the procedure with the duplicate bottle. Thereby, continue with the conduction of the test as planned. Use syringes fitted with needles of appropriate length to collect the liquid samples. Carefully invert the serum bottle and insert the needle through the septa to collect the liquid sample. Pull the plunger slowly to avoid the intrusion of gas bubbles during sampling. Place the cap on the needle and then remove the needle from the syringe. Fix a 0.45 micron pore size filter on the syringe and filter the sample into a labelled sampling cup. If the filter gets clogged, replace the filter and continue with the filtration until reaching the minimum required volume to perform the analyses of interest. Take 1 milliliter of the filtered sample and transfer it to a labelled cup containing 9 milliliters or 0.5 moles of sodium hydroxide. This sample will be used to carry out the analytical determination of the sulphide concentration dissolved in the water sample. Close the sampling cups and store the remaining samples for the determination of other parameters such as COD, sulphate, acetate and other volatile fatty acids. After 20 minutes, stop the shaker. The desired concentrations of lactate and sulphate required for this test should be prepared in advance and kept in 50 milliliter beakers. Using a syringe, add simultaneously 1 milliliter of each solution to each of the serum bottles. Once all the solutions have been added to all the bottles, start the stopwatch to monitor the time. Start the orbital shaker and incubate the bottles. Continue with the collection of gas and liquid samples according to the design of the experiment. In this example, stop the shaker after 15 minutes. Only take the first respective serum bottles for sample collection. The rest of the bottles can remain on the shaker. First, collect 3 milliliters of gas using a syringe and close the valve. Collect the liquid samples by slowly inverting the serum bottles. Store the samples in a sampling cup for further analysis. At the end of the test, stop the shaker. Remove the last set of serum bottles. Collect the required gas and liquid samples for the corresponding analyses. Remove the last sacrificial bottles and collect the required gas samples as shown previously. Thereafter, using a vial decapper, open the serum bottle and the septum. Pour the contents of the bottle into a beaker and mix the contents well using a magnetic stirrer. Under mixing conditions, collect the liquid samples in triplicate for the determination of the concentrations of mixed liquor suspended solids and mixed liquor volatile suspended solids, and other parameters of interest of the liquid phase. Continue with the collection of the required samples. Once all the samples have been collected from all the serum bottles, label the samples properly. If needed, transport the samples to the laboratory in a dry ice box. This test is performed in an anaerobic reactor. The results of the COD, sulphate and sulfide analyses of the samples collected can be organised and displayed using the working sheet suggested. The mixed liquor suspended solids and the mixed liquor volatile suspended solids concentrations should be monitored during the start and the end of the test for the determination of the maximum specific conversion rates. The volumetric sulphate reduction rate and the specific sulphate reduction activity can be estimated according to these formulas. As an example, a typical profile obtained from an SRB activity test is presented here. The slope calculated in this example is 225 milligrams of sulphate per hour, which corresponds to 2.34 millimoles of sulphate per hour. The amount of biomass in the reaction vessel was 4.5 grams of mixed liquor volatile suspended sludge per litre. The working volume of the batch reactor was 2.5 litres. Therefore, the actual biological sulphate reduction rate was estimated as 0.53 moles of sulphate per gram of volatile suspended solids per hour.